Does anybody want to go first, or do you want me to call on you? Elena? Okay, so Elena and then Amelia.
Oh, I can't share yet. Oh, there we go. Okay.
I just fixed it, yeah.
Thank you. You guys can see the presentation, right?
Yes, we can see it in speaker. We can see, yes, we can see the sides of the slides too, like in speaker view. I don't know if you meant to do that yet or not.
Okay. And then can you see my notes like in the Word doc or not? No.
No, we don't see the Word doc.
Okay, perfect.
But we see that, yes, perfect, that's it. Can I ask you, did you work on this with, thanks, Linda. Did you work with this, Elena, with Professor Serafin?
No, this is just research that I found. So it's actually part of when I was doing my reading for my thesis, which is about animal self-consciousness. I came across this study and thought it was really, really cool. And honestly, just really cute too.
They are
cute. So I felt like it would be fun to present because it's not something I've seen in any class before either.
So Professor Serafin, who's new this year, this is some of the research that she's done in the past and that she's planning to do. So I just was curious, so that's cool. All right, go ahead.
Okay, I think it just stopped sharing by accident. Let me try that again. Okay. No. Okay, all set. So I'm presenting today on the neuronal signature for monogamous reunion in the prairie vole. So again, like last time, I did a little bit of an outline of where we are throughout the presentation, a little bit of an overview, then going through the introduction, the methods, the results, and the discussion questions. And Professor Austin, I have some questions throughout. Should I ask to answer them at the time or just kind of keep moving through them?
I mean, we do have more time today, but I'm always nervous to make sure everyone gets a chance to go. So why don't you pose the questions, let people think about them for a second, but we won't actually take the time to answer them.
Yeah, definitely. So first, an overview. So who is Zoe Donaldson and why is her work important? So Zoe Donaldson actually has focused on studying the emotion of love and the neuroscience behind it for her entire career. In her undergraduate studies, she looked at voles and kind of what kind of bonds they can form and the way they interact with one another. During her PhD, she looked specifically at mice and actually created transgenic techniques to be able to manipulate neurons and their activities. And then in 2016, she started a lab at the University of Colorado Boulder and has been developing and using new optical techniques to look at neuron firing in voles when they're interacting with one another. So a little bit of an introduction. This is straight from a paper that she actually published in 2020. So why use voles as a model for systems of love? So she kind of talks about a lot. There's two different species of voles that she looks at. First, prairie voles and then meadow voles. So what's really interesting is that prairie voles actually are monogamous, but meadow voles she describes as more promiscuous. And so this kind of sets up this really nice dynamic where she can observe differences in their brain and kind of think about what kind of explanations might arise in terms of the behavior that she can observe. And just the question that I was going to pose is, why should we even care about the emotion of love and the way it is demonstrated by voles? So what is really important for this study is to understand what a pair bond is. And so it's defined as a selective desire to seek out and interact with a bonded partner. And this desire is maintained and reinforced over time. And so what she says is it kind of can be used as a proxy behavior for the kind of emotion of love that we as humans feel. And it's really interesting because actually only 8% of mammals even demonstrate this kind of pair bond. So it's a really small realm that we're a part of, and funny enough, voles are a part of. And the image here is a partner preference test, which is important to study a pair bond, and I'll talk about how that works in the methods. And so what do we actually know about pair bonds and what remains unknown and that will be attempted to be answered in the study that I'm talking about here? So What we already know is that the nucleus accumbens plays a primary role in encoding pair bonds, but we don't really know the neuronal dynamics underlying this and how that might change as the bond progresses. And so the primary goal of this study is to gain insight into that neuronal activity and to look at the formation and maintenance of pair bonds in prairie voles specifically. And so a question I had, because I know a lot of us had methods and things like that, is how do you think this might be accomplished in the lab? What kind of methods would you go about if you were doing this research? But then of course we can look at what she did in terms of methods. So she really employed two techniques here. The first is partner preference tests, which kind of attempt to answer the questions of whether a vole wants to spend time with their pair bonded partner or if they'd rather spend time with a novel vole. And so on the left, you can see what they do is they actually tie up a partner and the novel on each side of this box. And then the vole of interest is allowed to roam freely, and they kind of time which side of the box they spend the most time and how close they are and things like that. And during that time, at the same time, she implemented in vivo calcium imaging, which is where a mini probe is really put into the brain, specifically in the nucleus accumbens, and then you can see in real time what neural activity is happening. I'm sure we all know, but in vivo calcium imaging measures changes in intracellular calcium as a proxy for neural activity, so that's how that works. And then I pulled some images here from the study paper.
So cool. I'm sorry, that is really cool.
Yeah, I thought it was interesting, and I thought specifically the picture on the left is really interesting to kind of see what that would look like. So essentially they have that in while they're running around, and that's how she captures real-time data on that. And so there were some really interesting findings that she found. So the first is that voles actually do show a selective preference to interact with their monogamous partner compared to the novel one, and this gets stronger over time. So as the bond pair gets stronger, the desire to be with their partner also gets stronger in voles. And so the graphs here that you can see look at the total proximity, so the total amount of time they spend on which side of the box, the average interaction time, and then also the physical distance that they were from that person or that vole. And so the partner clearly, as time went on, they spent more and more time total proximity there. Also, their interactions were longer and they were much closer in that last graph. And so the other thing that they found from the in vivo calcium imaging is that there's a subset of neurons, which she calls the approach neurons, in the nucleus accumbens, which become very much more active when they are approaching their partner compared to the novel vole. And so here you can see again that in terms of activity, they look at the proportion of cells, which is graphed on the y-axis, and then you can see with the novel partner, the proportion of cell decreases, whereas for the bond-paired partner, that increases, which just means that more neurons are firing, the network is getting bigger, and the bond is getting stronger. And so in terms of discussion, this understanding is really important to provide insight into other types of relationships and bonds within voles. And so another question that I had was, do you think this research provides any insight into human relationships and bonds? And the interesting thing is that we already know, yes, it does. Actually, I think most of us have heard probably of oxytocin, the love hormone. And this was actually primarily found and studied through voles. So we already have this really big marker that has come from research with voles. And so this continued interest in the emotion of love through studying vole partner bonds is really interesting and very relevant to our daily lives. And so just a couple other questions that I had at the end. What implications do you think these findings about approach cell activation has for the understanding of human relationships? Can anyone really identify limitations of this study or things they would change in terms of methodology? And do you think there are other animal models which could be helpful in studying love, or do you think it's sufficient to study it in voles? So those were my questions.
That was really great. And I think the vole research is so interesting, like you said, because we have the two different sets of voles that are alike in so many ways and have this behavioral difference. between pairing and not pairing. And so I've read a lot of the work comparing them. I just think it's fascinating.
Yeah.
And they're these like little fuzzy, adorable things.
I would have never guessed that they, like, I don't know, I think I always knew penguins were monogamous and swans. I think those are like the prominent examples.
Right.
But I really didn't think that little voles would have that kind of emotion, which is why I thought it was really interesting. But.
Yeah, exactly. Any questions for Elena? Or comments? Okay, so I think I love that folks going in the chat. That makes my life so easy. But I think that Amelia had her hand up at the same time as Elena, and I sort of went to Elena. So Amelia, do you want to go next?
Yeah, I'll go next, and
then Linda, and then Amy, and then Sierra, right?
Okay. Is that working?
Yep.
Okay.
Perfect.
So I did my final presentation on the effects of cannabis on the brain. So just to introduce a little bit about cannabis, also known as marijuana, it contains the component delta-9 tetrahydrocannabinol, also known as THC, which is the primary psychoactive ingredient in cannabis. And often the potency of cannabis is evaluated based off of the THC concentration. Additionally, cannabidiol, also known as CBD, is often a subject of scientific investigation. And this is known to have a protective effect against the negative psychological effects of THC. And the available forms of cannabis are oils, herbal cannabis such as leaves and flowers, and hash. And also synthetic cannabinoids have been on a substantial rise. They also provide psychoactive effects, and but they can also have potentially dangerous substances. So that's a concern. So the evolution of THC and CBD ratios between 1995 and 2014, there's been an 8% increase in the average THC levels based off of a study done in the United States. And the CBD concentration also has decreased from 0.5% to less than 0.2%. And this could have a potentially negative effect because since THC is the primary psychoactive cannabinoid, and then CBD has a protective effect against the negative psychological effects, we could see more harmful effects rather than the positive effects of cannabis. So cannabis in the world has been slowly legalized in many countries. So on the left shows a map of the recreational use. It's still illegal in most of the world. As you can see, the yellow, blue, and pink are all illegal, and then the blue is where it's slowly being legalized. In the United States specifically, there's been 10 states have legalized its recreational use, and 32 states have legalized it for use for medicinal purposes, which is on the right. Here is the medicinal use map. And 45 countries across the world have been legalized medicinal use of marijuana. Okay. So I looked at a specific study focusing on medicinal marijuana use and chronic pain. So previous studies have demonstrated improvements in pain following short-term medical cannabis use, suggesting that there could be a chance for medicinal use to alleviate symptoms associated with chronic pain. And in this study, it was an observational and longitudinal study examining 37 patients with chronic pain evaluated prior to the initiation of this treatment. And it looked at the points 3 and 6 months after MC use. And the following symptoms were assessed, including pain, clinical state, sleep quality of life, and conventional medication. So MC patients reported significant improvement on several measures of sleep, mood, anxiety, and quality of life. And we can see these positive effects after 3 months and increased and strengthened even more after 6 months of treatment. So notable improvements in pain also can be observed in the decreased use of opioids after the 3 and 6 months period. So opioid use declined by 13% after 3 months and 23% after 6 months in these 37 patients. And also looking at a couple other surveys that surveyed dispensary members in New England found that 76.7% of 215 regular opioid users reduced their use of opioid medication slightly or a lot after initiation of MC. And similarly, in another study that surveyed MC patients in California, reported that 97% of prescription opioid users strongly agreed that they decrease opioid intake when they use MC. Additionally, the results revealed that increased THC exposure appears to be the most closely related to improvements on pain, whereas CBD exposure was related to improvements in mood, but not pain directly. So cannabis on the developing brain. As we know, the adolescent brain is dynamic and there are many structural and functional changes in the brain that occur well into young adulthood. So many health professionals see the need to inform public health policy pertaining to cannabis use in the youth. So there's evidence that there may be deleterious effects on neurodevelopment and which can result in poor neural health outcomes in adolescence, but there are other determining factors such as severe use histories, mental health, or psychosocial factors that can play a large role in determining these outcomes. So there's a lot of like mixed literature on the findings of this. Neuroimaging studies reveal that there are structural and functional differences in adolescents reporting a history of cannabis use compared to minimal to no use histories. But it's also found that some individuals may be more vulnerable to cannabis-related effects than others. But overall, the findings suggest that the risk of adolescent cannabis use outweigh any potential benefit during typical development. And then this was just another study that showed over increased lifetime marijuana use, there is also association with increased cortical thickness of the left entorhinal cortex, even after 28 days of abstinence. And the THC is thought to accumulate in neurons and it can become neurotoxic. The THC usually binds with the CB1 receptors, which can be found in higher densities in the hippocampus. There's evidence that changes in the hippocampus, cerebellum, prefrontal, and lingual regions were associated with THC and cannabidiol levels, suggesting toxic and protective effects, respectively. And then finally, I looked at cannabis and addiction. So cannabis is the most commonly used substance of abuse in the US after alcohol and tobacco. It's found that chronic cannabis use is associated with the downregulation of the CB1R THC target receptor, and they are restored after 4 weeks of abstinence, which is interesting. In addition to changes in reward processing, chronic cannabis use seems to affect emotion processing. This has been revealed after several MRI studies reveal functional and structural differences in the amygdala, which is a key brain structure in processing emotions. And one of the most consistent neuroimaging findings in addiction is that of dysregulation of the frontal cortical regions involved with executive functioning. Imaging studies investigating brain glucose metabolism, which often serves as a measure of brain function, reports decreased frontal metabolism in cannabis abusers when compared to healthy controls. So that's all I have. And then I just had a few discussion questions, but I feel like we've been skipping over these. And then here's my references.
Great. Go back to your questions for a second, just so people can ponder them for a second. I mean, I think especially the question about adolescents and even younger kids, like in states that have made recreational marijuana legal, some of the edibles look like candy and there have been kids overdosing. So I think it's an important question. I think we're going to move towards legalization, but I think we need to be thinking about these kind of issues. Did you look at all about the link between adolescent cannabis use and development of schizophrenia? It's something I'm curious about, but I haven't been following carefully.
I kind of looked at it. I feel like there's so much contradicting things out there, and a lot of with that especially there was a lot of news articles so that like completely misconstrue all the data found in the study. So I kind of just
Gotcha. Yeah, I'm curious about it. I keep meaning to look it up again, but I haven't looked at it in a while. Well, thank you. Are there any questions for Amelia? Nope. Okay. Applause. All right, so it looks like Linda's next.
Okay, so I'm sharing my screen now. Just give me a sec. Are you guys able to see the presentation?
Yes.
Okay, so I'm going to be doing my presentation on the adverse childhood experiences on the brain. So just an overview of what I will be presenting on. So I'm going to be talking about just a little bit of background about what it is and some facts that are some facts about it, and then what happens to the brain when someone is exposed to adverse childhood experiences, and then some treatments such as like meditation that is found when someone who experiences as ACEs, what happens to the brain when someone is practicing meditation with it. So just some background. So it's been found that people who experience ACEs were exposed to various types of childhood trauma. Some of the traumas that are included in ACEs is neglect, abuse, maltreatment. And then, so it has been found that there has been various effects on parts of the brain, such as functionally and structurally. And then almost 60% of Americans have experienced or been exposed to at least one type of ACEs, while 20% have been exposed to 3 or more. And then when a child experiences ACEs, there are difficulties with stress, emotion, behavior regulation, and reward learning, which could result in higher risk of psychiatric disorders, interpersonal problems, and engagement in high-risk antisocial activities. They found that 1/3 of people with mental disorders in the population were caused by exposures to childhood adversity. And just a little more background, it's been found that people who experience adverse childhood experiences, they're more likely to experience higher risk of late onset mental disorders. This can include anxiety, depression, schizophrenia, Alzheimer's, substance abuse. And then they found that meditation actually can be a useful treatment to help reduce the amount of stress they undergo. And this can help reduce the amount of activation in the brain associated with stress and emotions, such as the amygdala. And then, so I'm just going to talk about how it affects connectivity in the brain. It's been found that the VMPFC, which is the ventromedial prefrontal cortex, and the amygdala have been associated with adverse child experiences. There's basically like circuitry changes when they're exposed to violence or maltreatment or abuse. And it serves as a regulatory function over emotional responses by sending signals to the amygdala and the subregions of the amygdala. Basically, children have been found to have adult-like negative connectivity between the 2 brain regions when they witness a threat-related stimulus, such as fearful faces. And studies have also found that the amygdala is highly sensitive to early life stress, especially when children are exposed to ACEs at such a young age. And then, so this is one of the fMRI that was done on 57 adolescents during an emotional regulation test. It was found that there was a greater negative functional connectivity between VMPFC and the amygdala when viewing negative compared to neural images. Exposure to abuse was associated with more negative task-related function connectivity of left amygdala with both VMPFC subregions as compared to the control groups. And then now I'm going to be talking about like the brain structure and function that's associated with ACEs. It's been found that the brain regions that are affected by ACEs are the amygdala, hippocampus, anterior cingulated cortex, and the prefrontal cortex. Like I said in the background, there's been association with higher risk of anxiety, depression, and substance abuse. So I just want to talk a little bit about one of the studies I read. This was done on 20 patients who were diagnosed with schizophrenia, and they were compared to a control group of 20 healthy volunteers. And basically the study found that the patients with chronic undifferentiated schizophrenia were more exposed to ACEs than the 20 healthy volunteers. And then people with greater exposure to ACEs showed decreased responses in the amygdala and hippocampus while having increased activity in the PFC and ACC. They also found that the patients who were more exposed to ACEs showed larger gray matter volumes in both the PFC and ACC. This study concluded that alterations in brain structure and functions are associated with both chronic undifferentiated schizophrenia and the amount of ACEs exposure. And then lastly, like I've also read about other studies of children who experience early adverse experiences such as maltreatment have increased risk for memory problems, learning problems, and slower cognitive processing, which could lead to problems in school learning. So these issues in children due to ACEs result in a high risk of behavioral problems such as ADHD. And then so on the right is just a figure that shows the brain imaging of both white and gray matter in children. Basically, when they're exposed to ACEs, there's a decrease in cortical gray matter volumes and reduced thickness throughout the cortex. The top shows what happens to the white matter, and the bottom shows what happens to the gray matter. And then, so I looked more into like this treatment that's been done on people who have been affected by ACEs, and meditation was actually one of the treatments that they are currently studying. And it's been found to help mental disorders such as anxiety, depression, and others by reducing the amount of stress and emotion. This is done by helping reduce the stress on the amygdala. And it has also been found to increase cortical thickness in the hippocampus. Basically, when someone practices meditation, especially during stressful situations, there's a stress reduction in areas of the brain that are associated with stress. Over time, this can actually lead to a decrease in brain cell volume in the amygdala. And if the stress is reduced, which could lead to a decrease in fear, anxiety, and stress. And then meditation has been found to alter brain regions that are associated with learning and memory and areas of brain that are associated with emotion regulation. And then these are just some of the questions I had. And then and these are my references. I can go back to the discussion questions if anyone wants to answer them.
Yeah. Are there different experiences that are worse than others, or it's just like any of adverse experience tends to lead to these issues? Like the big teased out stuff?
I haven't read more into like which one's worse in terms, but I know that the more they're like, so the more they're exposed to ACEs, such as if they're like, there's maltreatment, there's violence and stuff more in the household, there is more higher risk of them being more depressed, anxious, and more like problems in school and learning abilities.
Right. All right. Thank you.
Like at certain ages also, like depending on what age they are when they're exposed to it, sometimes the development is worsened when they are exposed at a younger age or at a certain age.
Yep. Any other questions for Linda? All right. Nice job. Very nice job, everyone, so far. So Amy, I think you're up.
Yes. Can everyone see my presentation?
Yes.
Okay. So in my presentation today, I'm going to talk about the science behind hugs, as well as the impact that the pandemic has placed on our mental health. and how hugs can be beneficial towards that. So as we know, the COVID-19 pandemic, as well as the resulting economic recession, has been negatively affecting many people's mental health, and it also created a whole new situation for those already suffering from mental illness, such as depression and/or anxiety, as well as those with substance use disorders. Some of these negative impacts include difficulty sleeping or eating, increases in alcohol consumption or substance use, and the worsening of chronic conditions. And this is all due to worry and stress over COVID. As the pandemic continues on, it exposes many individuals to experiencing situations linked to poor mental health outcomes, such as isolation and job loss, among many other things. In this figure here, you can see that a majority of adults say that stress and worry related to the pandemic has had a negative impact on their mental health. This is up 14 percentage points since May and up 8 percentage points since the previous high of 45% in early April. And so I have some graphs here. In the first figure here on the left, it explores mental health and substance use during and prior to the COVID-19 pandemic. It focuses on populations that were particularly at risk for experiencing negative mental health or substance use consequences during the pandemic, including young adults, people experiencing job loss, parents and children, communities of color, and essential workers. And as you can see, there is a significant increase in reports of anxiety and depression from 11% to 41% from these populations collectively. Throughout the pandemic, anxiety, depression, sleep disruptions, and thoughts of suicide have increased for many young adults. They have also experienced a number of pandemic-related consequences, such as the closures of universities, transitioning to remote work, and loss of income or employment that may contribute to poor mental health. KFF analysis of the Household Pulse Survey finds that throughout the pandemic, a large share of young adults ages 18 to 24 have reported symptoms of anxiety and/or depressive disorder. It is 56% as of December 2020 compared to older adults, as shown in the third figure. And then many people across the country have experienced job or income loss, which has generally affected their mental health as well. Adults experiencing household job loss during the pandemic have consistently reported higher rates of symptoms of anxiety and/or depressive or disorder compared to adults not experiencing household job loss. So 53% versus 32% respectively. Findings show that households experiencing income or job loss are significantly more likely to report that worry or stress over the pandemic has negatively impacted their mental health. So throughout this pandemic, we've all been required to maintain a 6 feet distance from others at all times. Obviously, this is to limit contact with others and therefore minimize the spread of COVID. But when physical contact becomes limited, or in some cases eliminated, people can develop a condition called touch starvation or touch deprivation. When someone is touch starved, it's similar to someone who's starved for food. Their bodies are craving for food, but they can't eat because it's unavailable to them. And in this case, someone's psyche and body may want to touch someone, but they are unable to because of the fear associated with the pandemic, whether it be a handshake, a hug, or even a high five. Touch starvation increases stress, depression, and anxiety, which triggers a cascade of negative physiological effects. In response to stress, the body releases cortisol, which activates the body's fight or flight response. In turn, this increases heart rate, blood pressure, respiration, and it can also suppress the digestive system as well as the immune system. So this leads to an increase in the risk of infection, especially when it is chronic. People who are stressed or depressed, perhaps because of the lack of touch, will have problems sleeping, as mentioned earlier. Every single medical disease, including heart attacks, diabetes, hypertension, any single physical disease can be exacerbated if you are chronically anxious or depressed. And according to the latest CDC guidelines, we're at that point where fully vaccinated individuals can go ahead and socialize with others. They can finally hug their family members and friends without wearing a mask 2 weeks after everyone has received their second dose. So we're definitely getting closer and closer to the light at the end of the tunnel, but who knows if we'll even be able to go back to the pre-COVID times ever, really, which will be interesting to see. Anyways, now that hugs may start making their way back into the world, I will talk about the benefits of hugging. So hugs will activate oxytocin, which is sometimes called the love hormone, as Elena mentioned during her presentation. It is critical for bonding, especially between a mother and child at birth. Research has shown that oxytocin also affects our general well-being, enhances relationships, and induces calmness. In other words, it is involved in increasing positive feel-good sensations of trust, emotional bonding, and social connection, while also decreasing fear and anxiety responses in the brain at the same time. Hugs will reduce the stress hormone cortisol and improve mood. So there's a release of oxytocin, dopamine, and serotonin when presented with a hug, assuming that the individual finds that the stimulus is pleasant. Once the hormones are released, there's a reduction of stress, anxiety, depression, loneliness, isolation, and sleep disturbance from an improved mood. There's also an appraisal of emotional support, which will provide positive coping behaviors when experiencing negative emotions or events. So skin is the largest organ in the human body, covering us all the way from our head to toe. In a 2018 study by scientists at the Medical College of Wisconsin in Milwaukee found that skin communicates positive and negative touch stimuli to our sensory neurons. The outermost layer of our skin, called the epidermis, is predominantly made up of billions of keratinocyte cells. The keratinocytes release adenosine triphosphate, ATP, which then activates receptors on the sensory nerve to relay sensory information, such as physical touch, to the brain. When we hug or feel a friendly touch on our skin, our brains release oxytocin. As mentioned earlier, it provides us with a feel-good sensation. Hugging can also help our bodies fight off infections and therefore boost our immune system, according to a 2014 study conducted by researchers at Carnegie Mellon University. So to understand what's going on when we hug, we need to step back to our primate past with monkeys and apes. Monkeys and apes create and maintain their friendships through social grooming. As useful as grooming a bee for removing debris from skin and fur, its real effectiveness comes from the slow leafing through the fur that is involved as the groomer searches for foreign matter. We still do it up to a point. For example, when our parents leaf through, our hair when we were younger. And of course, this is also why we may find the attentions of our hairdresser to be quite relaxing. The slow stroking involved in grooming stimulates a particular set of nerves known as afferent C tactile neurons. They're only found in hairy skin and are quite different to the usual nerves that convey information about touch, pain, and pressure. These neurons respond only to light and slow stroking, and they have a direct route into the brain where they trigger the release of endorphins. Endorphins are neuropeptides, so small molecules that are used by neurons in the brain to signal to each other. They're part of the pain control system, and they produce an opiate-like analgesic effect. As a matter of fact, they're closely related to opiate drugs like morphine on a chemical level, but differ in two key respects. So number one, they are 30 times more effective as painkillers than morphine, and we also don't get so destructively addicted to them. And so here I decided to add a quick run through of a study by Rogers, Drill et al. To help reinforce the benefits of hugging in the long term. Here they talk about how humans have an innate need for direct physical contact and how the act itself is a source of interpersonal support and affection, which can result in feelings of positive affect, empathy, and catharsis. In the study, they examined the relationship between the availability of hugs and self-rated health in individuals aged 65 years and older. It is suggested that the need for touch may actually increase with age, but the opportunity to be touched is lessened. So as we age, the use of social networks begins to diminish, which leads to increased rates of social isolation and loneliness among older adults. So the exploration of the relationship between hugs and health in later life is especially important. This study analyzed secondary data from the Canadian Community Health Survey, which is a cross-sectional population level survey that collects data from community dwelling individuals. For this particular study, we mentioned that the target subjects were of the ages 65 and older. The measures in the study included self-rated health and hug availability. So self-rated health is a sensitive indicator of overall health and wellness, as well as disease risk. So this measure can be used to examine the relationship between the availability of hugs and overall health and wellness among older adults. This measure was assessed using a 5-point response scale with the question, in general, would you say that your health is poor, fair, good, very good, or excellent? The responses were then split up into 2 groups. So one group represented higher SRH, which included excellent, very good, and good responses. And the other group represented lower SRH, which included fair and poor responses. So the availability of hugs was the main exposure variable, and it was also optional for the participants to respond to. And so those that did respond to it were asked the following question: how often do you have someone who hugs you if you need it? And so the possible responses included all the time, most of the time, some of the time, or a little time, or none of the time. This study provides evidence for the role that hugging has on health and well-being in later life by demonstrating a positive association between the availability of hugs and self-rated health among adults aged 65 years and older. Based on the data, it was found that those who responded with all the time, most of the time, and some of the time in the optional questionnaire reported higher self-rated health than those who reported hugs available to them none of the time. In other words, this supports the health promoting role of hugs, as those provided with a wider availability of hugs were much more content and therefore had better health outcomes, as opposed to those who weren't provided with any for a long period of time. So this study established a therapeutic hugging week program for residents in a long care facility. Here they observed individual differences in the preference for receiving hugs and the number of hugs preferred among older residents. And while not everyone finds hugs pleasant or even comforting, they may value their privacy and personal space and may very well be content with a more limited approach to hugs. So it can't be assumed that older adults want or even need a hug. It also can't be assumed that older adults not in isolation have hugs available to them. And because the data is self-reported, it excludes any objective measures of psychological or physical health. In terms of future directions, it would be critical to examine appropriate contexts, settings, and implementation practices for hugging interventions to avoid any external factors outside of what is being measured, to influence the data more precisely by excluding those who do not consent to hugs. It is also important to continue examining the health promoting and disease reducing effect of hugs. As mentioned earlier, as human social interaction and connection become more and more digitized, the availability and reception of hugs warrants attention. And then here are my discussion questions.
Oh yeah, these are interesting. I don't know. Yeah, I think that's an interesting question, whether we'll go back. People will be cautious for a long time. Any other questions or comments for Amy? All right, so
I guess I'll go now.
yep.
Okay, you guys can see my screen, right? Just not in presenter mode.
I can't see anything yet.
Oh.
Saw your cursor, but that was it.
Interesting. Let me try this. Can you see now?
I can't. Can other people? Is it just me?
It's just a black screen for me,
Yeah, it's just black.
but I see a cursor.
Right. I see the cursor on a black screen.
Okay. The same thing happened to me earlier. Does somebody else want to go, and then I'll log in on my iPad so I can share?
Yeah, sure. Vivien, do you want to go? Also, Sierra, if you want to like send it to me and I can share it if you can't get it to work.
I should be able to do it on my iPad.
Okay, great. Vivien, why don't you go ahead then?
All right, I can go. Can everyone see my screen?
Yes.
All right, so my presentation will be focusing on Clostridium botulinum toxin, which is also called Botox, and how it's being used in the treatment of many neuromuscular disorder. The picture above is how the Clostridium botulinum toxin type 8 appears when reconstituted and sold at Botox. All right, so we're gonna start with a question so that I can interact with you guys. So what do you think, how much of Botox could kill everyone on Earth? So you can react to that on Zoom. Yes, no, slower or faster. And it could be 4 grams, 4 kilograms, 4 micrograms, or 4 milligrams. Jeffrey said it's 4 milligrams. Kayla said it's 4 micrograms. Some people said it's 4 micrograms. All right. So we'll come back to that question later. Okay, so when most people hear the word botulinum toxin, many associated these with bioterrorism. And after the 9/11, the US public has become more aware of the aspect of bioterrorism, particularly evident in the fact that President Bush signed into the law of Bioterrorism Preparedness Act of 2001 in June 2002. So this presentation is geared to understanding what Botox is, how it works, and what it can be used for. I hope to dispel some myths going on about Botox and increase our awareness of its usefulness, considering it is one of the most deadliest biological toxin in the world. So here's my outline of my presentation. It will begin by surveying the history background of Botox, paying particular attention to the botulinum toxin, and the majority of the presentation will focus on understanding the molecular mechanism and effects of the toxin when produced in mammals, and how this toxin has been identified as a key neuromuscular therapeutic agent. To start with, as defined by the CDC, botulism is a rare but serious paralytic illness caused by a neurotoxin that is produced by the bacterium. This bacterium is called Clostridium botulinum. And botulism can present in three forms, mainly categorized by method of entry of the bacteria into the human host. So foodborne botulism arises from eating the food that contain the toxin. Infant botulism most prominently presented in the ingestion of honey, which contain the spores of the bacteria, and it germinate into the intestine and release the toxin. And the final type of botulism is wound botulism, when a wound becomes infected with the bacteria. So the toxin is categorized within the A category, bioterrorism agent, mainly due to its toxin's great impact.
Lethality.
Lethality, thank you. I will discuss later how this toxin is actually a poor choice for a biological weapon. So these experiments on botulism has been recorded as far back in the 10th century with the Emperor Leo VI forbidding the manufacturing of blood sausages due to the fatal muscle paralysis that's exhibited by those who consume such items. Some of the severe outbreak occur in the 18th century in Germany. So the figure on the right, Dr. Kerner, a German physician, published his first study of botulism in 1820, and is based on the experiments he conducted a multiple of lab animals as well as himself. He was able to deduce how the toxin interrupted motor signal transmission in the peripheral and autonomic system. So these experiments also provided him with the hypothesis that the toxin could be utilized for therapeutic use, such as lowering sympathetic nervous system activity associated with movement disorder. Dr. Kerner was indeed, I think, beyond his time because he proposed that the toxin was the biological weapon that has some sort of related impact. And the word in Latin, poctulus, means sausages. Next, so post-World War I and pre-World War II, the United States began intense research into biological weapons. A covert operation was constructed in which Chinese prostitute would assassinate high-ranking Japanese officer by botulism poisoning via ingestion of a gelatin capsule that has the toxin inside. So a large batch of capsule was produced, but when tested on donkeys and 100 survival rate was observed, 100%, the project was aborted. So it is still in current debate as to whether donkey are immune to the toxin or not. Maybe. The toxin was one considered as an ideal biological weapon because of its dose. So 1 gram can kill 1 billion individuals. So given that we have approximately 8 billion people in the world, we can consider maybe like approximately 4 grams or 4 to 8 grams can kill them all, can kill us all. But however, it is a poor choice of bioweapon because it needs to be ingested in sufficient quantity, and it has variable mortality rate, rapidly inactivates in water, and is not person-to-person transmissible. So, and it can be sufficiently treated by antitoxin composed of neutralizing antibodies. So this is a picture of the bacteria itself. It is part of a group commonly found in soil. It is rod shaped and it grows under low oxygen condition. This forms spores and allow bacteria survival in an inactive phase until the correct environmental conditions are encountered to allow growth. So under the condition that remains unknown, it releases the toxin, and the toxin is the main virulence factor of the bacteria, which can cause botulism. There are several classes of the toxin, from stereotypes ranging from A to G based on their variability in structure, target size, and toxicity. And all 7 types work by the same general mechanism of inducing muscular paralysis by preventing the release of acetylcholine from the presynaptic neurons at the neuromuscular junction. Since they differ in target, their individual effectiveness in inducing paralysis is variable. In my presentation, I'm going to focus on the botulinum toxin type A, which is the most potent of serotype, and therefore it has been used frequently in research and in therapeutics. Type A and type B remain the only serotypes that have been manufactured and approved by the FDA for commercial use. So most of the research from 1940s with this toxin has been focused on its roles as a potential biological weapons. It is one of the most toxic toxin known to human. However, it also shifted its potential use in human as well. It is of interest that since it is accepted as the toxin as a valid therapeutic agent for neuromuscular disorder. So the first work was done at Johns Hopkins, where the doses of the toxin was analyzed on hind limb paralysis in chickens. And the first human disorder to be treated with the toxin was strabismus, which is an eye disorder that has high reoperative rate. So does any one of you know what strabismus is?
I was actually born with that, so I do.
Did you have a surgery on it when you were young?
I had surgery when I was 2, yeah.
Yeah.
And I was really lucky that it held, but for a lot of people, they have to get more than one surgery or still do exercise. I didn't have to do exercises or anything.
You are one of the lucky ones. Right. So with the onset of treatment using this toxin for strabismus.
So hold on, I'll just tell everyone, strabismus is lazy eye. Basically, it's where the eye muscles don't have the eyes be controlled conjugate, so they're controlled separately and they don't come together.
Yep. So the muscle doesn't align into eyes. So other neuromuscular disorder was followed in testing whether this toxin can be a therapeutic treatment. So now we have gone over the historical background of the bacteria. We're able to connect the disease to the biological agent, but researchers still do not completely understand how the toxin is produced, transmitted, or taken up by nerve cells to generate the clinical condition of botulism. So this part will focus on what we understand about its transmission. So why is it understood that the bacteria grows only under anaerobic condition, making the foodborne route the most common for botulism? What signals for the release of the toxins still completely not understood. However, we do understand that the toxin, once it is released, it can act upon the neuromuscular junction. The journey to the junction still remains unclear, but the detailed mechanism of how the toxin occurs at the neuromuscular junction will be discussed in detail later on. So in this slide, we see how the toxin in blue, a non-covalent multimeric protein complex made its way to the nerve. So once the toxin has entered the lumen, the lumen of the gut, it crosses the membrane barrier via receptor-mediated endocytosis. And then in order to get into the general blood and lymph circulation, once circulating, the toxin exit but via an unknown mechanism and reaching the extracellular space where it mainly acts upon cholinergic nerve cells. So it must bind onto the plasma membrane on the nerve cell and trigger the receptor-mediated endocytosis. So it goes inside, followed by pH-induced translocation across the membrane. So when it is in the cytosol, it acts as a zinc-dependent endoprotease to cleave the target polypeptides that are essential in the acetylcholine exocytosis. So by blocking the acetylcholine release, the muscle paralysis occurs. Again, this is the mechanism action of the toxin. In figure A, which is a normal neurotransmitter release, is mediated by synaptic function. So the SNAP proteins, including synaptobrevin, SNAP25, or syntaxin, are essential in forming a complex to trigger the exocytosis of the neurotransmitter. Once it is exposed with the toxin, so the toxin comes in, breaks down these SNAP proteins, the acetylcholine or any neurotransmitter will not be released. Therefore, it causes muscle fiber paralysis. All right. So now that we have gone over the mechanism and the transmission, let's review its therapeutic use. So besides the initial research conducted for the botulinum toxin as a bioterrorism agent, it also can act as a local paralytic agent or therapeutic agent on multiple neuromuscular disorder and pain management. This is an example of how the toxin type 8 induces local paralysis. In using it as a local paralytic agent, the only risk or complication that tend to arise besides immune response reactivity to the toxin is local paralysis. You can see before injecting the Botox and after injecting the Botox, the wrinkles disappear because the muscle there is paralyzed. The toxin can also act as the therapeutic agent. And in conclusion, these studies has further supported that the mechanism of action in Botox on neurocell is highly specific. However, this specificity is important in treating a variety of neuromuscular disorder from wrinkle to hyperactive muscle and pain-associated disorder. So there are studies that have tried using Botox in reducing migraines. And these two topics are the current area of research in future FDA-approved treatment of the botulinum toxin. However, while there are many neuromuscular and pain-associated disorder treated with the toxin, the Botox injection, only 4 disorder remains FDA-approved for treatment. So while these injection reduces the overactive muscle and pain associated with such disorder, the effects are fleeting, which means they need repeated injection. The injection remains a less expensive alternative to surgery. However, it is not a permanent fix. So one of the discussion questions I have is whether choosing between cosmetic surgery and scar versus multiple Botox injection Is it worth it in the long run or in the short run? Is it ethical to do so? There needs to be a discussion topic on it. And also, it's very social constrained. More research needs to be due because there is a stigma of using a deadly toxin in the human body. Is it a good use? All right, in conclusion, the toxin is a great therapeutic agent in its treatment of various range of neuromuscular disorder. However, to further the use of the toxin as a first-line treatment, we need to understand its mechanism of release. And there are relative few impediments in therapeutic as discussed in the previous slides, but overall, the future in treating with Botox is very bright. And remember, sometimes wrinkles are not that bad. So thank you for listening. And with that, I'm happy to take any questions.
All right, wait, go back to the cartoon because I didn't finish reading. No, after that, Leonard.
Oh, this one.
Yeah. Gotcha. Any questions for Vivien? Really interesting. Thank you for that.
Thank you.
And all the kind of implications you made into policy and cosmetics. Thank you.
Yep.
Any questions or comments for her? All right, Sierra, do you want to give it another go?
Yep. I think this will work. One second. Can you see?
I see a thing that says you're sharing your screen, basically,
Okay.
but I don't see anything on it. Oh, I see a puppy. Okay, now I see cerebral palsy.
Okay. There we go.
Good.
Okay. So my presentation is on cerebral palsy. I am, let me see here. Okay, here we go. I'm first going to talk about what it is, then I will mention some of the different types. We'll get into pathology and diagnosis and some treatments, and then I have a couple discussion questions as well. So first off, what is cerebral palsy? So cerebral palsy, or CP, which is what I'm going to refer to for the rest of the presentation, is a group of non-progressive brain injury or malformation disorders that vary in severity and affect normal movement in different parts of the body. And so the word cerebral refers to the brain's cerebrum, which is the part of the brain that regulates motor function. And the term palsy refers to describing the paralysis of voluntary movement in certain parts of the body. It won't go next. Oh, here we go. Okay. So some of the types. So this diagram's a little bit confusing, but I think it does a good job breaking down all the different types. So spastic cerebral palsy involves tightness of the limbs, and it's caused by damage to the brain's motor cortex, which controls voluntary movement. And it is also caused by damage to the pyramidal tracts, which help relay signals to the muscles. And so sometimes people might refer to this as pyramidal CP instead. So that's kind of like the same thing. Hemiplegia affects one side of the body, including the arm, leg, and trunk. Diplegia affects either both legs or both arms, and quadriplegia affects all four of the limbs. Dyskinetic CP involves excessive involuntary movements and can either be athetoid or dystonic. Athetoid CP is characterized by a combination of hypotonia and hypertonia, which is loosened muscles and then stiffened muscles, which causes muscle tone to fluctuate. And this type of CP is caused by damage to the brain's basal ganglia. Dystonic is alternating between rigidity and flaccidity. Dystonic CP is a movement disorder that's marked with hypertonia, increased tone, and uncontrollable involuntary posture and movement. Dystonic CP often interferes with the child's sleep due to uncomfortable pain caused by the contractions, and can also be difficult for children with dystonia to sit or lie down. And lastly, ataxic CP involves the cerebellum. Ataxic CP makes up about 2.4% of all CP cases, and this type of CP causes ataxia and issues with balance, coordination, and voluntary movements. So pathology. The mechanisms leading to CP can be attributed to many factors, but I kind of just highlighted 3 broad main factors. So the first one is intrauterine exposure, including infection, elicit a fetal inflammatory response syndrome, and white matter damage in the fetus. It also causes fetal growth restriction, placental vascular disorders that are associated with vascular injuries in the fetal brain, and congenital anomalies. Another one is intrapartum events that affect the fetus during the process of labor and delivery, such as birth, asphyxia, and placental abruption are all acute events that are associated with further implication of the fetal brain. And last one, postpartum exposure to infection of prematurity complications that affect the neonate and subsequently lead to CP. These events can be isolated but also combined, and a neonate who suffered from hostile intrauterine environment, such as infection, may be further affected by acute intrapartum events such as abruption or postpartum complications and develop CP. Diagnosing. So most children with CP are diagnosed during the first 2 years of their life, but if a child has mild symptoms, then it can be difficult for a doctor to make a reliable diagnosis before the age of 4 or 5. And so doctors will order a series of tests to evaluate the child's motor skills. During regular visits, the doctor will monitor the child's development, growth, muscle tone, age-appropriate motor control, hearing, vision, posture, and coordination in order to rule out any other disorders that could cause similar symptoms. And although symptoms may change over time, CP is not progressive. If a child is continuously losing motor skills, the problem is more likely a condition other than CP, such as a genetic or muscle disease, metabolism disorder, or tumors in the nervous system. Lab tests can identify other conditions that may cause symptoms similar to those associated with CP, as well as neuroimaging techniques that allow doctors to look into the brain, such as MRIs. They can detect abnormalities that indicate potentially treatable movement disorders. And some other neuroimaging techniques include cranial ultrasound, which uses high frequency sound waves, computed tomography, which uses X-rays to create images that show the structure of the brain in the damaged areas, and MRI, like I said before, as well as electroencephalograms, which, as you guys all know what that is. And here, this diagram on the right is just illustrations of severe injury on the top row and subtle injury bottom row in cases of cortical malformations in the first, which is the first column, white and gray matter lesions, middle column, and ventricular enlargements in the last column. Treatment options. So the most common one treatment option for CP is physical therapy, and it usually begins in the first few years of life or soon after the diagnosis is made. Specific sets of exercises, such as resistive or strength training programs and activities, can maintain or improve muscle strength and balance and prevent any contractures. Also, another treatment option that is pretty common is like orthotic devices, like special braces, to help compensate for muscle imbalance and increase independent mobility. And braces and splints are used to correct these abnormalities and improve the function, such as just simple actions as sitting or walking. And other orthotic devices help stretch muscles or the positioning of a joint. So braces, wedges, special chairs, and other devices can help people sit more comfortably and just make it easier for them to perform daily functions. And so CP is caused by the lack of oxygen to the brain during birth or pregnancy. And this lack of oxygen causes damage to myelin. And when it's damaged or destroyed, the nerve cell decays and eventually dies. And so a new treatment option, stem cell therapy, that has been in clinical trials, has been used, and it's supposed to, the stem cell therapy creates the opportunity to take the stem cells and manipulate them to replace the damaged myelin and protect the neuron from dying, thus avoiding further brain damage. And so this form of therapy is still in clinical trials, but is showing promising results. So I have a couple discussion questions. How do you think cerebral palsy compares to other neurological disorders in terms of prevalence? What preventative measures would be most effective, and how can we better educate people on these measures? And how do you think CP affects adults? And these are my references.
All right, this is great. Thank you. Questions for Sierra? Any questions or discussions about any of the talks we had today? Y'all are just tired, right? You just can't do it anymore. All right. Thank you. These were great. I thought all the presentations were just fantastic. That's everybody, right? Everyone. All right, good. Fantastic. All right, good luck with the rest of your finals and projects and everything. And hopefully I'll see most of you at the senior dinner. We'll be in 2 groups. And thanks. Good luck towards the end of the year, and I'll see you at commencement after that. Bye.
Thank you.
Okay.
Thank you.
Yep.
Thank you.
Thank you.
Bye. What's up, Rachel?
Hello. I don't know if you saw my email this morning with the review sheet.